Okay, so just a quick recap of glycolysis. Remember that in glycolysis, ultimately glucose is coming in. This process takes place in the cytoplasm. So when glucose comes in, we need to kind of lock it in place and get the ball rolling here. So we're going to use two ATP molecules. So two ATP come in and it helps with the conversion of glucose to fructose diphosphate. Because we invest some energy here, we say that this is the energy investment step. Fructose diphosphate then gets split into two G3P molecules. Each G3P is going to be converted to pyruvate, and as that process happens, we get out two ATP here, we get out another two ATP here, and we get out two NADH molecules. Remember that up until this point, our net yield of ATP after glycolysis is only two molecules. Even though we get out four, two of the four are going to cancel out the two that we needed to use initially. So that's why we only have a net yield of two. After glycolysis takes place, we then, what happens next is determined on whether oxygen gas is available. So we in science say the fate of pyruvate depends on the availability of oxygen gas. Since oxygen gas was available last time, I talked about the entire process of cell respiration, aka aerobic respiration, which was going through this transition step, the citric acid cycle, and then ending with oxidative phosphorylation. But now we're going to look at not this process, we're going to look at what happens after glycolysis when oxygen gas is not available. So the fate of pyruvate is going to be anaerobic respiration, which is also called fermentation. And there are two basic types of fermentation. There's alcoholic fermentation and there's lactic acid fermentation. These two processes are very simple compared to true aerobic respiration. So here we are. We're going to look at alcoholic first. Both processes take place in the cytoplasm of the cell for those particular organisms. So in alcoholic fermentation, here we are at the end of glycolysis. We have created pyruvate. Pyruvate now, because oxygen is not available to be the final electron acceptor, what happens now is pyruvate is going to get converted to alcohol. The alcohol specifically would be ethanol. Okay, so it gets converted to alcohol, and then as a byproduct, we get carbon dioxide gas, which is released. So in this process, this is carried out by some plants, carried out by yeast cells. They could survive on this. Ultimately, what we see here is that we're not producing in either type of anaerobic respiration. We're not producing any ATP. Where the ATP is made is back here in glycolysis. So we're going to keep on getting out those two ATP molecules. And because microbes like bacteria and fungi, like yeast cells, are very tiny, these ATP that are being generated during glycolysis as a net yield is able to sustain them. However, us and other more complex organisms like larger plants and animals and other fungi need something a little bit more complex than this. But what this does do, if we go back to both types of respiration, is it regenerates the electron carrier molecule NAD+. And as long as we're regenerating NAD+, so NADH is coming in from glycolysis, it's dropping off those electrons and the hydrogen ion there, and that's helping with drive this process to convert pyruvate, to this case, an ethanol and CO2. And we regenerated that NAD+. So that NAD+, as long as it's being regenerated, can come back in to glycolysis, pick up electrons, pick up the hydrogen ion, and we reform NADH. And NADH could come back in. So as long as we have NADH coming in and NAD+ being regenerated, this whole flow can continue. But once we stop producing NAD+, this process would shut down because we'd get a buildup of electrons. So, as far as alcoholic fermentation, we get ethanol and CO2. This is carried out by some plants. We see it happening in yeast cells as well. As far as the food industry, if you should ever take Food Micro, it's alcoholic fermentation. That's going to give us our alcoholic beverages, wine, beer, hardcore liquors. Alcoholic fermentation is also used in the bread making process. So when you take those yeast cells and you mix it in with the dough, you're going to let the bread dough rise for a little bit in warm conditions. So what's happening is those yeast cells are breaking down the sugar via glycolysis, converting it to pyruvate. We get out alcohol, and the carbon dioxide that's released is going to help the bread dough to rise. When we bake the bread, we're going to burn off that alcohol. So that's how we use alcoholic fermentation in the food industry. On the other end, we have lactic acid fermentation. Lactic acid fermentation, just like alcoholic fermentation, occurs when oxygen gas is not available or becomes deprived. So after glycolysis, each pyruvate molecule that's made, because remember after glycolysis everything happens twice, so each pyruvate is just simply going to get converted to lactic acid or lactate. And again, we have NADH coming in from glycolysis, dropping off those high energy electrons to help with the conversion, and we regenerate NAD+. And as long as NAD+ is being regenerated to go back into glycolysis to pick up those electrons, this process will continue. Lactic acid fermentation itself, it occurs in bacteria. It occurs in some animal cells. For example, our animal cells, or our cells, during a strenuous workout, will convert over to lactic acid fermentation when oxygen is being depleted. And ultimately, that's what leads to the burning sensation in our muscles during a high-intensity long workout, such as if you're doing weight training or running a long-distance marathon. You get that burning sensation in the muscles. Ultimately, what would happen then is lactic acid would be transported via the blood, and it would go to the liver, and the liver, which its major role is to detoxify, is going to break down lactic acid back into pyruvate. So these are the two basic types of anaerobic respiration. Depending on the organism, will determine which type of anaerobic respiration takes place. And the key thing there is that we really don't generate any ATP, but what we do generate in this process is that NAD+, which could go back into glycolysis to keep that process going to produce those two ATP molecules. And for a bacterium and a small organism like a yeast cell, those two ATP are sufficient enough to help do the help carry out the work or the cellular functions needed for those particular cells. However, a large organism that is more complex like ourselves and other plants, we need to have true aerobic respiration take place. So in closing, there are two basic types of anaerobes. We have obligate anaerobes and facultative anaerobes. Obligate anaerobes, as their name implies, they're obligated to anaerobic conditions. So these organisms thrive in oxygen-poor environments. And in fact, because they're so obligated to being in an anaerobic condition, if oxygen gas is available, often these obligate anaerobes will die off in the presence of oxygen. That's not the case, though, with the facultative anaerobes. Facultative anaerobes thrive in both aerobic and anaerobic conditions. So they'll still thrive and are able to convert between the two processes, whether oxygen gas is available or not. So that ultimately is our lecture on anaerobic respiration. Going through all that, this finishes our studies for Unit 3.